Good morning, everybody. My name is Morten Sandset, and I'm Vice-Rector at the University of Oslo. And I'm very happy to welcome you all to this morning event, where we will present to you a few of the research projects that have been developed and focus on different aspects of COVID-19 infection. COVID-19 is the cause of a terrible pandemic that we could not have imagined only 1 year ago. It not only causes severe infection, that is sometimes fatal and more often is associated with long-term sequelae, but it influences daily life activities and social interaction. Many people are just suffering due to the infection itself or due to the many social impacts. If there is one positive outcome of this disastrous pandemic, it is that outstanding research in science again has gotten the respect and appreciation it should have. There is no doubt that cutting-edge research and innovation has been key to handling the pandemic. And I believe the standing of science and researchers in the general population has increased considerably. In fact, when trying to get a full overview of all the research related to and focused on COVID-19, we had a hard time. There were just too many, including the development of vaccines, but also the research on how the pandemic impacts us, tools for tracking the virus and to prevent spreading, and also research on how our economy is affected. Personally, I recently got involved in the project comparing the effects of heparins on the outcome of infection in Finland and Norway, due to completely different policies for treatment in the two countries. Strong and resourceful research groups are imperative for our societal preparedness when facing crises like this, especially those we are not yet foreseen. Within weeks of the outbreak, scientists from the University of Oslo were hard at work, some from their temporary offices at home, some while homeschooling their children. For that, we as a society should give thanks and show appreciations. So today's Impact Breakfast is one way to show off all the insightful and important work done by our scientists. I'm eager to hear today's speakers. We will have 7 presentations, total 15 minutes, including questions and answers. And you may give your questions, upload your questions to the speakers during the presentations. And we will ask the speakers. It is now my pleasure to introduce the first speaker, who is the section leader of research services, Toit, from our IT department, who will speak to us about our service for sensitive data, TSD, a platform for research and innovation with sensitive data. Leon, I cannot say the floor is yours, but the screen is yours, please.
Thank you, Per. So I'd like to tell everyone a little bit more about the University of Oslo's services for sensitive data. And by doing that, I hope that you as a researcher or a company know more about what we can offer for you. So research in the time of COVID-19. Now, we are a research service provider, so we don't do research ourselves, but we've had a lot of interesting and meaningful collaboration with various parts of society that are involved in research. And 2 of the most high-profile cases have been the so-called Corona study and the TRAN study, which has been done by researchers at Oslo University Hospital. And these studies have been really successful in having large-scale surveys and have had very good response rates and also found very interesting results. And TSD, we have also helped TV2 to do a study where they looked at the impact that COVID-19 has had on people's social lives. And then also there's a European project called EASK Life, with whom we are collaborating to establish a repository for clinical trial data for COVID-19. Lastly, we've had very tight collaboration with the National Public Health Institute, also known as Folkhälsoinstitutet. They are involved in the Corona study and Tran study, and we are also in discussion with them about helping them to scale up their operations regarding the sequencing of viruses and transferring this data from secure zone at Oslo University Hospital to a secure zone at Folkhälsoinstitutet. So I'd like to tell you a little bit more about the Corona study, about its size and about how we managed to help researchers to get it done. In total, there have been more than 120,000 unique respondents to the survey over several waves. And getting participation rates like this is very difficult, and also maintaining such participation over several waves is really difficult to accomplish. But the combination of features that we have been really instrumental in doing this. So first and foremost, we are able to collect consent from people before they start to answer the survey. Most importantly, this consent is digital. It has to be digital, especially in this age. And it has to be dynamic, meaning that if you consent to something now, you should have the opportunity to revoke that consent in the future. So having consent as digital and dynamic means that it gives stakeholders insight. So the university hospital can request insight into who has consented to what study. Researchers themselves can see who has consented to what and use that in their analyses. And all in all, this increases transparency in the research process and helps people to comply with the requirements of the GDPR. Now, collecting several waves of results from more than 100,000 people can give you a quite difficult data management problem. And that is why our platform generates data sets automatically for people in well-known formats such as comma-separated files, SPSS, and Excel. This means that once your survey has been completed by all respondents, you can log in and start analyzing. You don't have to collect all of this data into an analysis-ready format. It is already done for you. Also importantly, the results are anonymized. Now, this doesn't mean that we don't collect the personal information about people, but it means that we split the data set into two. So one part of the data set contains personal information, and that is only accessible by project administrators, while the anonymized part of the data set is accessible by everyone and used for analysis. And then importantly, this whole process is self-service, and of course we add user support to that because sometimes there are difficult decisions to make in survey design, and we try to help the researchers as well. So I've spoken a little bit about research during COVID-19 and the Corona study specifically, but I'd like to pause and tell you a little bit more about the services for sensitive data that we actually have. I know there might be listeners in the audience that are doing all kinds of research, and this is therefore relevant. So the services for sensitive data is a secure cloud and a remote desktop, meaning that you get a virtual machine, you log in and you do your work. You bring your own computer, you bring your own data, and you do your computing. As for computing, we have advanced computing services, high performance computing, graphics processing units for machine learning, field programmable gate arrays, specialized for genome sequencing, containers, custom hardware, pretty much anything that you need will make it happen. I've already spoken at length about the survey offering, but if the survey functionality that we have is not good enough for you, then we also have a platform for app development. We offer advanced user support. Many of our staff have PhDs themselves and years of involvement in research. And the platform now hosts about 4 petabytes of data, serving almost 50 different institutes across Norway. It is an extensible platform. And if we look at the graphs that I've plotted here, we see the total amount of projects that have used us since 2014. And now we're up at 1,200 active research projects. We see a similar growth in the amount of researchers that use TSD, now being up above 6,000. So our approach is that we offer services for the entire data lifecycle. We have services that help you get started and administer users and project parameters. When you're done with that, you can move on to data collection and or app development. For tablets, you can use online surveys. We have our own data portal for uploading and downloading files, and also for recording from webcams. And soon we will launch screen recording as well, which can be very useful in times where you have to do interviews over Teams or Skype or Zoom. Having collected your data, you of course need to work with it and analyze it. And this is where we have computing services, software services, very importantly, along with the storage, you get backup of your data so that your data doesn't get lost. And we offer a variety of databases if you want to organize your data in a database. An important part of research, of course, is sharing data and publishing, which can be a challenge if you're working with sensitive data. But we have developed secure services for sharing data, and most of the time this would be anonymized or sharing an individual's data with themselves. So if you have a patient, for example, and you want to send a report to them, we have our publication portal where you can do such things. So I'd like to just show you what it is like to use TSD. What would you see if you were doing research there? You would either see a Windows remote desktop or a Linux remote desktop. And as far as that is concerned, this is just another computer where you work with your data, with the added benefit of your data being secure. In addition to that, we have a host of web services and APIs, and the API is an acronym that appears more and more these days. It is an acronym for Application Programming Interface, and what this means is that all of our services are available from programs, not just from web browsers. So in practical terms, this gives you the opportunity to make your own services and customize that to your needs. We also have command line tools, mobile apps I've already mentioned, and very importantly, user support throughout the entire journey. So you can find us by Googling TSD UCIT contact or by following this link. And if you start a dialogue with us, if you read our documentation and you figure out that we don't yet have what you need, then all you need to do is to talk to us and we can make it happen. Thank you.
Thank you, Leon. This is very impressive, and no doubt that this service has been very instrumental to all the research that is being done currently, also particularly with COVID-19 infection. So now, Angelique, do you have any questions from the audience?
Hi, no, there are no questions so far from the audience.
Anyone in the team that will like to address? No, that's quite clear then, Leon. So
I hope that's good news that there are no questions.
No, but I mean the service is fantastic. I mean, and it's absolutely necessary for the research that we're currently doing in the field. So could you say a little bit about the functionality? I mean, there's been some scientists think it's a little bit difficult to work within the TSE room.
Yeah, working with IT can be challenging in itself.
Yeah.
We also find it challenging sometimes. And I guess shortly I can say that is why we invest so much effort into user support and helping people with everything from logging in to installing custom software to optimizing software, procuring hardware. We really take a holistic approach in helping people, and we take great pride in doing that.
And also accruing the data with mobile units, I guess, is probably exploding these days.
Yeah, it is a challenge to keep up with all of the possibilities and requirements, but that's why we've built a platform that we can extend, and we try to at the same time cover 90% of the needs by default, and then cover those specialized cases when they do arise. And we're happy to work with any research team on covering these cases that we can't currently do.
Okay, Angelique, any new questions arising from the audience?
Hey, no, not so far either.
Okay, then we say thank you to you, Leon.
Welcome.
Thank you very much. And then we go on to the next speaker, who is Professor Kristin Braa from our Department of Informatics. And she will speak to us about their district health informatics system, which is now used for COVID-19 surveillance all around the world. This innovation is a global significance, of course, and have not gone unnoticed. And only a few weeks ago, our Crown Prince Haakon asked for a meeting to hear more about the system and its benefits. So now it's your turn, Kristin. The screen is yours, please.
Super, can you hear me?
Yes.
Oh, good, because I don't see any mute button here, so I'm all by my own and my screen. So good morning, everyone. I'm very happy to be part of this Impact Breakfast Seminar, talking about our work on DHIS2, which has become a platform for digital public good. And I will tell a little bit about the story behind, but hopefully talking about how we do local innovation and the global response to the COVID-19 surveillance and pandemic. The pandemic has shown us how important it is with data for decisions. And even in Norway, we know how much importance there is to have good quality data, to have targeted innovation and intervention, I will say, not innovation, sorry for that. So this screen, this first picture here is actually from a local innovation on the border of Uganda to Kenya, where all the landlocked Uganda, all the truck drivers were on the border in order to be able to wait for their certificate or for their negative result of their COVID test. So here it's a local innovation, making an app on Android, as you can see on the right side there, directly linked to the lab result and printing a certificate. So the truck drivers were not standing on the border for longer time than necessary and be able to in a safe way, come into the country with goods. This we don't even have in Norway as of today. You know that, I guess. Wow, now it's not next here, so I'm trying. There it is. The story behind, I don't have time because I only have 10-ish minutes, but it's a long story behind this health system information program. It started as a post-apartheid program in 1994. The most important thing is it's action research. So Leon was talking about tools for research. This is actually tools for operational systems in countries where we are doing research on how to make it sustainable, how to make it scalable, and how to build capacity to use local innovations. So it has traveled through geography, like in South Africa, post-apartheid, traveling through all Africa, Asia, Middle East, Latin America, to also traveling through geography, technology, from Microsoft Access to now using all the open source and APIs that Leon was talking about, all the technical facilities in order to cater for the low resource context in the global south. Meaning you can use Android and you can do having offline support even though you don't have internet at hand. So we have been a collaborative center for some years. So we are for WHO. So we are working very closely with WHO and all the global health partners. So this has becoming a partnership, resulting in over 70 countries using DHIS2 for all HMS, health information systems programs within the countries, within the ministries. Okay, so it's a global public, becoming a global public good. That has been possible. There are many stories to it, but one of the stories is, of course, it needs to be funded. So it has becoming a partnership between all the global health agencies, with NORAD in the forefront, supporting us already from 1994, with the PEPFAR, which is the presidential US government presidential effort combating HIV/AIDS, and the Global Fund, CDC, which is the American Public Health Institute, UNICEF, Gates, and so forth. This graph is only to show that it's correlated 1 million of Norwegian kroner. We are now 125 million per year. So we say 1 million per country. It's just a little bit fun part that you need to have someone to fund in the platform in order to be able to become a global digital public good. But back to the action research, which is the most important. So how has it been possible for a university-led program to cover Global South with a health information system platform? It's because it's action research. We have built building knowledge while we build systems, and we are building capacity, meaning we have 65 PhD graduated through this program, and 25 is active. We actually have a defense coming up on Friday from Dar es Salaam, Tanzania, and he's heading the HISP in Tanzania. So what we do, we build capacity for local to support the region and to support the countries. And again, in addition, we have international master programs. And the master students of Department of Informatics is also taking part of this. Don't have time to talk about all the other academies we have. So this rocket you can see there. So what we do is actually local innovation. When we have a problem like the pandemic, like the COVID-19, we are actually doing an innovation, solving it on the ground. And whenever we can be able to solve it in a low resource setting, it's coming into the machinery, outcomes capacity, meaning guidelines, implementation guidelines for this highly configurable platform. How do you configure it in a robust way in order to be able to adopt this local innovation? And whenever the next release comes, it becomes part of the software, so all the other 70 countries can use this innovation. So here, this is a very complicated, very big lot of text, but you can see here the open source development and implementation is done in the global community. So it's not only HISP at the University of Oslo, which I led, which I'm leading. It's actually the 15 other HISP groups, and you can read all of them because I don't have time to note them. They are regional, and they actually, again, build capacities in countries and supporting the countries. And who are funding? The global health agencies are funding these HISP groups through University of Oslo, where we help to look into global repository, building up all the global resources needed in order to help the countries. So this collaboration we also do with the WHO in order to disseminate the standards that the WHO have developed through many years in order to, because of this highly configurable platform, the health information system can be designed bad and good. And then the health apps are then helped in order to do it in a good way. So all the global health agencies are funding, but we are then implementing the best practice standards. And you can see here as a kind of downloadable on HIV, TB, malaria, morbidity, immunization, vaccination, mother and child health, and so forth. So what happened then? So when the 27th of January, Sri Lanka got the first suspected cases, 2 cases, and they are a tourist country. They know how important it is to protect the border for the virus like the COVID-19. So 2 days after, they were able to configure a port of entry app on top of the HS2 in order to trace the incoming tourists from China through their travel while they were tested, through their travel inside Sri Lanka. And you see here, this is actually for a long journey, but they actually have made apps for all these kind of full stack processes, from port of entry to quarantine centers to hospitals, community response, and ICU bed tracking. And this led to the global response. Because of these 15 HISP groups are collaborating together, this innovation in Sri Lanka spread immediately to all the other HISP groups, and we made global resources in order to support this effort. It was a hackathon in March where all these many of the apps were built. It was supported by the software team in University of Oslo and become available. And in March, 12th of March, WHO declared it as a pandemic. But already in April, we published, well, in March actually, all the COVID-19 packages. Why I showed the packages before is we also made packages for the COVID-19, and then it will become easy for all the countries that already had DHIS2 to be able to implement COVID registration, port of entry, contact tracing, and so forth. And here you can see the various digital health data toolkits, case-based surveillance, plus laboratory, point of entry screening, contact tracing, outbreak line listening, and daily reporting. So this has been worked with all these 15 HISP groups together with WHO and the team in Oslo, together with all the researchers in the HISP contact network. And in May, this was the headline in the Aftenposten. So Norwegian contact tracer use pen and paper. In Uganda and Sri Lanka, contact tracing is carried out much more effectively using digital tools. How is this possible? So that created a lot of fuss. And today, we started the work already in March, actually, to work with Tromsø and Asker. And today, through the fixed platform in KS fixed platform, 100 and 110 municipalities are using DHIS2 in Norway. But in contrary to the rest of the world, DHIS2 is 1 instance per municipality. So the design of the decentralization in Norway is actually not working for our benefit when it comes to having solid data, because it's actually up to all municipalities to make their own solutions. That's very strange. So the rest of the world is have 1 instance in the ministry. In Norway, 1 in each municipality. Local innovation. Coming back to the local innovation that is so important in order to respond quickly, because this pandemic, we know how fast regulatory bodies are changing their regulations. We know that from every week we hear Anna, or maybe every day we have new regulation in Norway even. And so the response on innovation. So Leon was talking about the APIs. The DHIS2 is highly configurable and is open APIs to be able to do apps on top of this platform. And this innovation was this printing of travel pass, but also contact with the labs. And this innovation then will be able then to be copied around the globe. And the next, what we are doing now is, of course, we are coming into the vaccine. You maybe have read that the Africa is not really high up in the queue for vaccines. However, we are preparing the DHIS2 in order to be able to help the countries in order to deploy the vaccines in a sustainable and good way. And then it's built on established approaches and partnerships in the countries, which we already have with the Gavi, the global vaccine alliances. Already many countries, 28, are using DHIS2 for vaccination of children. So we learn from that one. And of course, we have local expertise and innovation. So this is already launched, and we have a public webinar this, it's actually tomorrow, on Thursday, tomorrow. So it's actually where we are training people. We have had internal webinar, but we are training people in how to deploy this app for vaccination. So in order to end my presentation, so what has been the response from the HISP network? We actually have leveraged of the DHIS-based health information systems in countries, and as well as the capacity. The most important thing is actually the capacity. Like Palestine suddenly sent us a beautiful use case in May, where they say, we have now been able to implement DHIS2 for COVID-19 surveillance in the Palestine, based on Sri Lanka's work, and we didn't even know it because they just the HISP Sri Lanka helped them, and they had the capacity from before because they had used already DHIS2 in the whole country. We of course have learned from Ebola. We were part of the Ebola work as well, and then there we saw that strengthening existing systems is the most important thing. You cannot sit down and learn and implement things in a pandemic, because the response time is super important. And of course, we have leveraged on the collaborative network. As you can see, all the HISP groups that are already there, contact with all the ministries. They were also in lockdown, so they used WhatsApp, Zoom, all the digital tools, all the digital resources that we have. We are a collaborative center, so of course, the work together with WHO, UNICEF, CDC for standard-based approach is super important. And leveraging and building up global digital resources available for everyone for free. That's the base for becoming a global digital public good. It's for free, but nothing is for free. So someone needs to fund somewhere, and we got additional fund from NORAD already. 1 week into the lockdown in Norway, end of March, we got 35 million in order to be able to speed up the whole network to be able to be responding to this pandemic. And we are regarded as one of the one in the CDC. I had a presentation presented as the one that we responded to the pandemic in the fastest way. And we have a 5 years collaborative agreement now with the CDC in order to be able to prepare for the next pandemic, or of course live with this pandemic, because it doesn't seem like it will go over very fast. Now we are into the vaccine, as well as monitoring adverse event, meaning side effects of the vaccine is of course also very important. So I think I will end there. Thank you so much, and I'm available for whatever questions.
Thank you, Kristin. This was really amazing to hear. It's always great to hear you talk about the HISP system. And I learned about the child and mother care system that you had, which was even better than our own in Norway. So this is really great. Are there any questions from the audience? Angelique, I encourage you all listening that you now have the possibility to ask Kristin questions and the next speakers also.
Nope, there aren't any questions from the audience, but just a little question from myself. What have been some of the challenges that you've met while trying to implement these? I'm from Tanzania, so I know that sometimes the infrastructure can be really difficult to implement. So I'm really curious as to how you managed to implement this in such an effective way, and what are the challenges that you've had with that?
You might say that the mobile revolution has revolutionized also our work, because we have used mobile internet, and you actually get mobile internet everywhere-ish. But because we have developed this offline support, meaning you don't need to be online all the time. That's very important. So yeah, web-based, but also the capacity in country and the long-lasting relationship with the ministries. Because what we do, like in Tanzania, they are our friends since we have collaborated with them since 2010. 7, 6. For so many years, we have worked with the ministry in Tanzania. They are our best friends. They have teams inside the country, and they can utilize all the innovations. And they build the procedures and institutions around best practices. So it, as much as Per Morten would know, innovation is only monitored whether it's used or not. So we have solidly focused on the deployment and the use of the system. So that is kind of, I would say that that's a network, that the action network effect, that we actually capacitate people to own the system and be able to respond to changes that are needed in order to make the system relevant always, because you need to redesign, make it relevant. And this pandemic shows that we need to be very responsive in order to be able to meet all the new requirements.
Thank you.
Questions?
I could just to get an idea is, I mean, all these countries and units that are collaborating, but what about the hardware? I mean, is it cloud-based, and do they interact with each other directly, or and what about the privacy, which is so important in Norway and other countries, Western countries?
Privacy is important everywhere. So that's what I'm saying. It's actually one instance in each country. So each country decide themselves whether they would like to have cloud-based, whether they will have a local host, or they will actually sit with the server inside the minister's office, which someone like, you know, then we try to convince them that maybe not so sustainable, because then we of course need to capacitate how to do this. So we have guidelines how to set up this in the most secure way as possible. So, and since PEPFAR is using it, the US government is using it for their HIV interventions in all their 58 countries at their production system, it's gone through all their security tests and licenses. So it is as secure as you design it in a way, which all systems are.
Yeah. Okay, Kristin, then we are on time. Thank you very much for this fantastic presentation. And then we go on to the next speaker. And I would say that there are few, if any, scientists that are more known for studying plague than the next speaker, Professor Nils Christian Stenseth, from the Faculty of Mathematics and the Natural Sciences, and from the Department of Biosciences there. And he will speak about his research project, the dynamics of the Covid-19 pandemic under external drivers at different stages. So Nils Christian Stenseth, please.
Thank you very much, Per Morten, and thank you very much for inviting me, dear friends of science. This project is really an epidemiological project. I myself, I don't think about myself as an epidemiologist. I'm a curiosity-driven science, evolutionary biologist and ecologist. And I, by chance, got into epidemiology. I was studying for many years the effect of climate variation on ecological systems, many different systems, and I got sort of bored of it. There was nothing new. So I asked people, I would like to study a system that has this reservoir out, a disease system that has its reservoir out in nature, but affect people tremendously. They said, malaria you should do. No, I said, there's so many smart people in malaria, so it's very difficult to make a niche within that field. And by chance, I got access to fantastic data on plague in Central Asia, specifically in Kazakhstan. Monitoring program since the end of the Second World War up until the breakdown of Soviet Union. Absolutely fantastic data. I started working on this in 2000 and had our first paper on that disease, the plague, in Science in 2004. And 2008, we published a review paper. It's a crazy idea to really have been in the field really only for 4 years and to publish a review paper. That paper is being cited very much by really all plague papers. Why? Because we came from outside and asked different questions than typically was asked there. Today, I'm going to talk about well, since then I've been working much on plague. I've been working on Hanta, dengue, avian flu data system, etc., etc. Now I'm going to tell you about this project of ours. Could you move to the next one? Which is the title of that project. I was actually in China the first week of January when it all started. The public didn't know that there was an epidemic starting, but I happened to have good friends, so I knew there was an epidemic starting. And I was, in a way, happy I got back from China on the 10th of January. In March, we submitted a proposal to the Research Council entitled what you see in front of you. And now I'm going to give an overview of that project and give some glimpse into 3 sub-projects within this overall project. Basically 3 papers that we have, 1 published and 2 currently in review. Could I have the next slide? This is an overview of the project. You don't have to look at the details here, but it's really a dream team. It's people from Norway, University of Oslo, the Veterinary Institute, as well as people from key places around the world. Penn State and Princeton are involved. And we are fortunate to have an advisory board of very centrally placed people around, be it in US, in China, or in Africa. Could you go to the next one? Well, it's divided up in 3 basically work packages. This is the first to sort of get an overview of the environmental factors, seasonality, climate variation. Much of my work has been on climate variation, as I said. Go to the next one. And then doing basically modeling. This is a core of my field, trying to understand the dynamics with external drivers. And I'm going to tell you about a project on that, or a suite of projects. Uta Bjørnstad, by the way, is an early student of mine. He has been listed in, I think it was A Magazine, as one of the most successful Norwegian outside Norway within science. Next one. And then the overall aim of the project is really to prepare ourselves for the next pandemic, really learn from past pandemics, be it the different plague pandemics, as well as the current one, to be better prepared for coming pandemics. So now I'm going to continue by giving a glimpse of what we have been doing within 3 projects. 1 of the papers are accepted and published. 2 are still in review. Could I go to the next one? I have very good connection in China, which is fantastic actually, because I get access to data from China CDC that most other people, I get easily access to data from China that other people might have to work much harder on. And this is a study out of Tsinghua, where I'm affiliated, Tsinghua University. But it's really based here in Oslo. With the first author, she is from China. She was with us for a while doing a dengue paper, then left us to Imperial College, now has come back and is working with us on this very project. RIIM Li. Could you go to the next one? Next one. Yeah. No, one back, please. The previous one. Yeah, stop there. This is a simulation study based on data from China and the rest of the world, simulating the dynamics in a proper way. And the overall conclusion is that if we are to reduce the effect of this disease, we really have to have a global action. And we have to target our action into some of the hotspots. In a global world, we predict here that we could reduce the pandemics after a couple of months if we close down some of the key big airports in the big cities around the world. And you see on the map to the left there where we have to act. Really taking this into the Norwegian setting now, a good strategy would be to deal with the pandemics where you have the hotspots, for instance in Oslo, but then having this coordinated so that we don't spread the virus around. Could you go to the next one? This is in review now in Science Advances, and you go to the next one. It's an age-structured. The next one. It's an age-structured SIR model, very standard, but we are then taking account of the age structure of the population, of the human population, as well as the contact structure between or within age groups and between age groups. Go to the next one. So, well, this is what we do for a living. So, Per Morten, as is, in charge of the university here, this is what I do for a living. And that's why I think you should continue to give me salary, because this is the stuff I'm doing. So the demography, the two figures on the top left there, this is demography. We take real demographies of countries, which is easily available, and then social mixing strategy. And you see the mixing with the ages. Most of them are interacting with their own age class, but there is some interaction with others. And this is different from country to country. Then we fit the parameters to the country we are dealing with, and then go to the next one. And we observe dynamics like this. It's sort of stabilizing after a while, but that depends on where we are. Go to the next one. Yeah. So we can ask questions, and we have in this paper, we have asked questions about what happened and is likely to happen within different countries. And we have countries for all over the world, 11 countries, and we have them listed on the right here. And the metrics is really sorry, I should go back a second here now. What we then are asking, what is the death rate in the population of the pandemic? And that is different in different countries. South Africa is less hit than many others because they have a very young age structure. Italy is harder hit, partly because they have a very old age structure. And then we can also modify the social mixing and then asking what kind of intervention strategies is the best one. We are using this model now for analyzing what should be the best vaccine strategy. The overall aim is to reduce the death rate, but it might not always be the best strategy. If you have limited vaccine doses to target the old one, maybe you should target the young one with a high high social interaction. Go to the next one and the final project. I said I'm a curiosity-driven scientist. And I'm an ecologist, and I'm doing quite a lot of ecological work, dynamics. And my very good friend, Steinar Engen in Trondheim, we were sitting chatting a little bit and said, how should we deal with import of virus when we really have control over the vaccine, over the disease? And go to the next one. So we developed a model based on stochastic age-structured Leslie matrix. This is a typical population dynamic model structure in ecology. And then really ask what kind of scenarios will happen when there are different rates of transmissions coming in from outside. I think this is a very helpful tool, not under the current situation, but when we have control and want to continue having control. Go to the next and final one. Thank you very, very much for your attention.
Yeah, thank you very much, Nils Christian. This is of course extremely important for understanding the development of the pandemic, and it was interesting to hear your comments on taking it back to Norway, and you said some words, but what would be your advice to the government if you were really to do one thing?
Well, the advice to the government would be to think, have a national strategy, and really the strategies should be decided centrally and should be coordinated so as to take hotspots in Norway and get control. So this decentralization is very dangerous, actually.
Yeah, I think we heard that from Kristin too. I mean, the decentralized tracking system are also extremely well, not very efficient, at least. Okay, Angelique, do you have questions from the audience?
Nope, there are no questions so far.
No. So also I learned the 10 key airports that you really could have taken down the spreading of the epidemic. That's also sort of interesting. So some key measures like that could have probably slowed down spreading of the infection a lot.
Yeah, what we're doing now, as I alluded to, we are asking if you have a limited amount of vaccine doses, how should you go about reducing the death rate? And that question will be different from the situation we are in now, and further on in a year, 2 or 3 further down the line. Should you really target vaccine first, the old and risk groups, or should you take the groups that are most socially mobile and have social mixing? You might think that the answer is obvious, but there is lots of indirect benefits.
Yeah. Do you have some data on that, or?
We are writing up a paper now.
But you want to share some of that?
I don't want to share this because it's, modeling stuff is very simple, but you have to do it.
Yeah.
I can share it in 3 weeks.
Yeah. Okay. We are looking forward to that. So thank you, Nils Christian. That was fantastic.
It's a great pleasure.
Yeah. Okay. We go on then to our next speaker, who will talk to us about how we can utilize modern data sources to inform about the spatial spread of the COVID-19 in Norway, and also to predict the number of new hospitalizations. And this is the presenter is Solveig Engebretsen, a research scientist from the Norwegian Computing Center and from the Norwegian Institute of Public Health. So now the screen is yours, Solveig, please.
Thank you. And good morning, everyone. I'm very happy to be here. Thank you for inviting me. I'm going to present the model that the Norwegian Institute of Public Health has been using for situational awareness and forecasting during COVID-19. And when you want to look at the geographical distribution of a virus, then mobility is essential because that's what drives the geographical spread. So that's how the virus arrives in a new location, either through local mobility, so mobility within Norway, or through mobility from outside, so imported cases of COVID-19 from abroad. But if you want to include mobility, then of course you need to have some data on how we move. And in Norway, we are very lucky to have good publicly available census commuting data from Statistics Norway. However, is it likely that we, during a pandemic, will move according to those census data? Most likely not, because the pandemic affects our behavior and also there are different restrictions. So now, of course, we have seen that the mobility has been affected by the pandemic, but we had to make that choice in February last year before we had seen any mobility data. But we decided that we wanted to include an alternative mobility source that could inform us about the real-time mobility during the pandemic, where they were thinking that the mobility will not be stationary, and B, the mobility will differ from non-pandemic periods. And luckily, we already had a collaboration in place with Telenor. So a little more than a year ago, I defended my PhD thesis, which was part of the research center Big Insight. It was a collaboration between the Norwegian Institute of Public Health, the University of Oslo, and Telenor. And in that project, we worked on how we could utilize mobile phone data to build models for infectious disease spread. And by using mobile phone data from Bangladesh, from Telenor, we built a model for influenza spread. And that's really the model that the COVID-19 model is based on. And in Bangladesh, the largest advantage was not really the real-time aspect, but more that there are no census data. So you need to have some alternative mobility source. So Telenor, they have location data on their customers in terms of which cell tower they are closest to. So for every 6th hour, each individual gets assigned a position. But those individual level data are not the ones that we use or have access to. So for privacy reasons, we use aggregated data. So we know the total count of individuals who have transitioned between any pair of municipalities in Norway in every 6-hour interval. There are no individual identifiers in the data. We don't track any movements. So we don't know if it's the same people who travel from Bærum to Oslo in the morning who also travel back from Oslo to Bærum in the afternoon, for example. So those types of rules we have to invent ourselves in the model if we believe in them. Here it's plotted 2 of those mobility matrices from February. To the left, we have individuals who have changed position between 2 a.m., where most of us are at home, and 8 a.m., where at least some of us are normally at work. And then to the right, we have individuals who change position between 2 p.m., where most of us are at work, and 8 p.m., where most of us are at home. So we see that most of the mobility links are short distance travel links, but there are also some very long range travel links here. And we also note that there are more people who are moving in the afternoon than in the morning. So how do we use these data in our model for the disease spread? The model is a so-called meta-population model, where we take the country and divide it into groups. So for the model that Nils Christian Stenseth just represented, you have age groups and disease, and here we have geography. So the groups are here municipalities. And then inside each municipality, there is a relatively simple model, mathematical model for the disease spread, where the virus can be transmitted between individuals who are present in the same municipality, with a probability that depends on the number of susceptible individuals, so healthy individuals who can contract the disease that are present, the number of infectious individuals, and the number of immune individuals, and also the infectiousness of the virus. So this reproduction number that's become very famous last year. So there's one of those processes in each municipality, but those processes are coupled through the individuals who travel. We start the simulations of the spread on the first day where there was a confirmed case of COVID-19 imported from abroad to Norway by placing an infectious individual in the residence municipality of that case. For that day, we also have mobility information. So we move people according to the mobility matrix of that day. Then we let everyone mix for 6 hours, where the virus can spread between individuals who are present in the same municipality. After 6 hours, we again have mobility information. So we move people around and so on. And that's how we model the spread of from late February until today. We also include into our simulation all the confirmed cases of COVID-19 imported to Norway from abroad by, on those dates, inserting infectious individuals in the municipalities they travel to. So for this infectiousness of the virus, the reproduction number, we calibrate it to data. So we assume that it's constant, but we've allowed it to change in different at different time points, corresponding, for example, to interventions, times of interventions. And then we choose the reproduction numbers. We calibrate it to data using a statistical method, making sure that our simulations are matching both the hospital incidence data and the number of confirmed cases for Norway. And what's plotted here is our simulations in blue and the data in red. And we see that we're able to capture the trends. We can also look at the actual estimates of the reproduction number in different time periods. And by comparing them for different time periods, we can say something about how the situation has changed in different periods. So for example, if we compare the estimated reproduction number before mid-March to the one after mid-March, when the semi-lockdown was implemented in Norway, then we see a reduction in 83% in the reproduction number. And more recently, we also had some interventions implemented on November 5th. So we can compare the reproduction number before and after November 5th, and then we see a reduction of 44%. And then the most recently estimated reproduction number here is acting from January 4th, where we again had many interventions. So these estimates are last week's estimates, and this week's estimates are soon ready. But we see that our most recent estimate is a reproduction number of 0.65, so below 1, which is an improving situation. We can also look at the mobility to see how that has changed during the pandemic. So what's plotted here is the amount of outgoing mobility for each county on each day. And it's relative to March 2nd. So it's set to be 100% on March 2nd, 2020. We clearly see for all counties here that we have a reduction in the mobility in mid-March. After that, there was a steady and slow increase in mobility all until summer. And each line here is a county. So this one, the red one here is Oslo, and this one is Viken, and this one here is Nordland, for example. So in summer, we see a clear summer effect, vacation effect, where there's reduced mobility in the largest counties. And then some other counties have an increase in mobility. So for example, Nordland. After summer, the mobility was rather stable for some time. And then we've had dips when we've had restrictions and also vacations. And we clearly see a large reduction around Christmas in mobility. Finally, we can also look at the number of foreign SIM cards roaming on the network of Telenor. So this is the number of foreign SIM cards present in every county. We here also clearly see a reduction around mid-March, and then an increase when the borders were reopened around in late summer. And we see also very low numbers during Christmas. And after Christmas, it's been increasing. I just want to acknowledge the whole team, because this is the work of the whole FHI COVID-19 modeling team, which is a collaboration between different institutes. So the Norwegian Institute of Public Health, the University of Oslo, Telenor, and the Norwegian Computing Center, where I work as a researcher. Thank you.
Thank you very much. Very useful research and the data. So now, Angelique, do you have any specific questions from the audience?
Hi, there aren't any questions so far.
No.
Okay.
Again, very useful data. So you see the movement, but what about the connection between the movement and the infections and the spread of the virus specifically? Can you say more about that?
Yes. So the mobility is very important in the beginning to say something about the spread, because in particular, if you have a disease where you only have one imported case from abroad, then you really need the mobility to see where it's spreading. If you have a high level of the virus, then it's a bit less important because then there are so many potential travel routes for the virus. And also if you have importations to many different municipalities, then it's also a different setting, of course. We have looked into the correlation between this estimated reproduction number and the mobility. We don't find a very clear correlation here.
Yeah.
Yeah.
So how would you translate it into if you were into a government or a health or protection advisor? How would you use that information?
So the mobility that we have here or in our model, that's mobility between municipalities and here between or out from counties. But there's also a very important part with the mobility within the municipality, of course. So how many social contacts we have that are not represented. So I would definitely not say that mobility reduction is not important. Because I mean, reducing mobility means that we have fewer social contacts.
Okay, thank you very much, Solveig, and we have to move on. And now we are going into diagnostics, and it's a pleasure now to introduce Professor Christian Braa, who's an old friend actually from the Faculty of Medicine, and who is now working in his own company, Vitas, in the Oslo Science Park. So you will tell us about developing a COVID-19 antibody test, Christian. So we're looking forward to hearing you talking. Please.
Thanks a lot. Can you hear me?
Yes, yes, we can. Yes, we can hear you. Please.
Yeah, very good. Thanks for the invitation. I'd like to see the next slide and tell you that Vitas is, I'm representing Vitas in this connection. This is kind of a strange story because it is a spin-off from the University of Oslo Department of Nutrition. And we have now 27 years of experience. And what is special to this contract laboratory is that we are able to measure almost any small molecule and even big molecules like antibodies in small quantities. And that has been our sort of concept all the way since we started. We now have a fair number of employees and a fourth story of the newest addition in the Science Park in Oslo. And we have about 50% more robots than we have employees. And we are serving different groups like pharmaceutical companies, biotech companies, academic institutions, hospitals, startups, and also multi-marketing companies. Next slide, please. When it comes to COVID-19, we know that this is a pandemic that is hitting everyone in the globe, and it's per yesterday 100 million globally infected. And about 2.1 million dead with the infection. So it's a big challenge. And as a laboratory, we were in a position where we could use our technology, measuring small amounts of compounds in the blood, on a dried blood spot technology that we have been using for about 15 years. Next slide, please. Next slide. And when you go to a regular office for blood sampling, it is expensive and slow. It is disease oriented. It's large volume of sample, 5 to 10 ml, and there are often no data interpretation or guidance related to the result of the analysis. With modern fingertip sampling on filter papers, you can get away with 50 microliters of blood. That is one blood drop. You can do this by yourself. You don't have to travel. You don't have to pay a lot. And analysis for almost any compound can be done on this small amount of blood, especially in relation to infectious diseases. It is very attractive because you don't spread your viruses around. When you dry the blood on a filter paper, it is considered being non-transmittable. The drawback of the system, though, is that you have to validate all analytes on this matrix. And that is what we have done for more than 200 different analytes. Also COVID-19 or SARS antibodies against SARS-CoV-2 virus. Next slide, please. Next slide. I'm not going to go into details here about the immune response, just point out that one relatively easy way to measure the traces of the virus in the body is to measure the antibodies that are relatively stable for a fair amount of time. So this is in contrast to the virus that is relatively stable for about 10 to 14 days in sputum, for example. The blood samples of antibodies left after the infection is much longer detectable in small blood samples. So that is what we have taken advantage of and developed the test. Next slide, please. Next slide. So we have done the validation, as you can see here on the right-hand side on the top. There is a correlation that is remarkably good between samples measured on the dried blood spot and samples in regular blood plasma. You can't get it better than that. And we also have stability for a long time, like 20 some days after sampling on the blood spots on the filter papers. So that is for the validation. And then we have tested on the next slide. Sensitivity and specificity when it comes to people infected with the virus, documented the infection. We very early after the virus came around in Norway, we were able to pick out PCR positive subjects, and we have followed them for 6 months, as you can see on the right-hand side. We have an increase in antibody IgG against the spike 1 protein that is on the surface of the virus. And there is an increase to about 6 to 8 weeks after PCR documented the infection. And then it is a fall off of antibody concentration of about 40% during the next 6 months. And these samples are taken from people naturally infected, and it would be nice to know if there is a different response when it comes to vaccination. Some data from the mRNA vaccines indicate that if you have 2 shots of vaccine, you have a higher level of IgG antibodies against the antigen given as a message RNA compared to a natural infection. Next slide, please. I've already told that it is important to verify that you have had an infection by measuring the antibody. So that's one good thing about getting this information. The other thing is that we have been collaborating with University of Tromsø, trying to describe the incidence of positive antibody levels in the regular representative Norwegian population by selecting 30,000 individuals and measuring the antibody levels before the vaccine came. So far, it doesn't seem to be a lot of individuals that are antibody positive, in spite of the fact that it has been documented about 60,000 people infected by the virus, according to PCR measurements. So I think it will be quite interesting to measure the antibody levels before and after vaccination to see if the antibody response is going to be, first of all, good with most people who will not have a positive response, and how fast will the antibody levels reduce after finished vaccination. And then it is all the time a question also of how long time does the immunity last, and what about the mutants? All this can be measured by our principle with using dried blood spots on filter paper, adjusted for antigen depending on what mutants we have. Next slide, please. Here is the instruction that is followed in every test kit that you can get when you buy or get a test kit submitted in the mail. Next slide, please. And here is the response letter, depending on what you have the answer is on your test. So you get the description of the result, and you get also, if you like, some more information about the virus infection and what in practice to do. Next slide, please. This test has been used in 2 big studies. The TRAIN study that included about 3,000 training members of an exercise facility in Norway, the so-called TRAIN study. And we have also, as I mentioned in the next slide, done a study, next slide please, done a national screening, the so-called Corona and Immunity in Norway with the Tromsø University. And the data are still being processed, and we have not got all the 32,000 samples yet submitted to the lab. Next slide, please. And then the last slide is to say that it is commercially available through different drugstores or pharmacies, and also certain web pages. And you can get the kit submitted via mail, or you can buy it on certain spots like the reception in the Science Park or in different drugstores, or you can get it from Body Markers. And you can get the results via Vita's internet page with the reports. One thing I would like to add is that it is a little bit disturbing to us that the government agencies have been sort of reluctant to collaborate on our technology, which is a little bit disturbing because we have the insight and the experience to measure in a very smart and inexpensive way things that is very complicated or impossible to do other ways. So we'd like to express that concern. Thank you for your attention.
Yeah, thank you, Christian. Very interesting, very important also to have good diagnostic tests. You talked about the sensitivity and specificity, and can you elaborate a little bit more upon that towards, more conventional antibody assays?
Yeah, our sensitivity and specificity is just as good as the regular test on blood plasma. So we have sensitivity of higher than 95%. That means every once in a while we have some samples, some people that have had their positive PCR documented SARS-CoV infection, whereas we can't find the antibody. On the other hand, the specificity is really high. We have tested 601 samples taken before year 2020, and none of them showed antibody against. So the specificity is really very high, 99%.
And also about, antibody reaction is of course important, but there is this speculation about immunity not being cell-based, and you cannot, and you still have immunity by not having the antibodies. Can you comment?
I think that is a biologically really important point. And so far, the antibody testing is the most convenient and relatively easy way to do it. It is several research-based other tests that can be used for testing, for example, cellular immunity, and that is much more expensive and resource requiring. So at the time, I don't think that is feasible on a large-scale basis. But it could be in the future, but that is requiring much more science.
There is also another question from the audience. Will you develop a test for detecting antibody in saliva also, looking for IgG and IgA?
I think, it's just a question of what people require or the demand from the market. I think we could do that because we have the sensitivity and we have the antibodies. So I think that is just a question of how useful will people or the medical society find it, and we can do it.
Okay, Angelique, are there any more questions from the audience? We could have one more.
And there are no questions from the audience so far.
Okay, thank you, Christian.
Thank you.
For the presentation. And then we move on more to treatment matters, and then Professor Hedvig Noreng from the Institute of Pharmacy, she will talk on the topic of medications' impact on the course of COVID-19. So Hedvig, please go on with your presentation.
Thank you very much. I hope you can hear me well. Good morning, everyone, and it is a pleasure to be here today and to talk about one of my favorite topics, and that is how we can capitalize on Norwegian registry data to answer important questions about medication safety. So this presentation will be about medications, it will be about COVID-19, but I also want to, in the sake of time, to focus on a specific patient group, that is the pregnant women. And I'd like to actually to start by asking the audience a question. And so please, if you are able to put your reply to this question in the chat box. So this is a question I got 2 weeks ago. It was a woman, she was a nurse, a frontline worker from the United States, and she got her first vaccine dose, the Pfizer vaccine, and a week later she discovered that she was pregnant. So she was early in her pregnancy, about 6th gestational week. So the question is, should she take the dose 2 or not? And there are 3 options. So please put your reply in the chat box. It's either A, no, she shouldn't, B, I don't know, or C, yes. And I'll actually ask Angelique to give me an overview in just a couple of slides, and then I will show you what the current recommendations are. Because these are important questions, and patients, they have lots of questions. And this is another one. This is from the Aftenposten, where the Norwegian Institute of Public Health says that we should wait to become pregnant after the vaccination. And there's actually so much different information out there. Some say, well, wait, some say 1 week, some say 2 weeks, some say it's difficult to plan pregnancies, so you just have to continue your life. And really information changes a lot very, very fast. And there's so many questions related to this specific group. For example, are pregnant women at higher risk of getting COVID-19? We saw from the influenza, from the swine flu, that actually pregnant women were more vulnerable. We are wondering about whether, well, if pregnant women get COVID-19, are they at higher risk of getting severe disease? Is there a chance of vertical transmission of SARS-CoV-2 across the placenta? And what is the impact of the disease on pregnancy outcome? And what I'm very much interested in is what is the impact of medications to treat COVID-19 and the vaccines on pregnancy outcomes. So there are very many questions that are still unanswered. We do have a little bit of information, and I'll just summarize. This was from a publication from the BMJ last year, where if we look at the sparse data that we do have on a little over 2,000 women, where when compared pregnant women with COVID-19 versus pregnant women without, the first finding was that we saw that pregnant women with COVID-19 were a little bit more likely to be admitted to intensive care treatment for COVID-19. That actually might be due to providers being concerned about the pregnancy and not the illness in itself. We do see the same kind of risk factors among pregnant women as in the general population. And there have also been signals of increased rates of preterm births. However, please bear in mind that this data, the majority of this data comes from China or the US, and it might be very differently in other parts of the world where the situation is completely different. And for example, here this is from a Danish study, they actually found that the rates of extreme premature infants had decreased significantly during 2020 compared to previous years. However, the reasons for this were unclear. And of course, this is something that we're very concerned about also on a European level. So once a week, every month, all 29 member states of the European Union gather at the European Medicines Agency. And in addition, there are representatives from healthcare personnel and patients and 6 commission-appointed independent scientific experts. And I'm one of those. And in this committee, which is called PRAC, which is the Pharmacovigilance Risk Assessment Committee, we discuss safety of medications and how to survey the safety of medications in Europe. And of course, we have not been able to meet physically. This is a picture from Amsterdam, where the European Medicines Agency is located. But everything last year has been like this. So we've been meeting virtually and of course discussing the COVID-19 situation in Europe, and the medications have been the top priority. And this is also why the European Medicines Agency funded this study that I would like to talk a little bit more about. This is a study where we're uniting all of Europe to try to understand how the COVID-19 infections in pregnancy and its impact. And we do that by using secondary healthcare data. And this we can't do in Norway alone. Although we have most beautiful registries that we can link, we do not have enough pregnant women. So in this project, we're uniting researchers across Europe to be able to cover 1 million pregnancies in 2020, and including the top 5 countries with COVID-19 in Europe. So that would be like Spain and Italy and Great Britain, and of course Norway. And it's being led by us at the University of Oslo, this work package. And this is the slide that shows you which Norwegian registries will be linked. And many of these will be known to you. You will see the data source 10, that's the Norwegian prescription registry. You will see, for example, data source 3, that's the medical birth registry. Data source 4, that's the MSIS that you all know so much about, where all the cases of COVID-19 are registered. The 6th SISVAC is our registry for vaccination. So because we have all these great registries that cover our entire population that can be linked on an individual patient level, it enables us to do really essential work. And for us, this is the first time we're linking actually 10 registries all together. So that's going to be a record for us. And of course, this is done in, it has been approved by REC, our ethics committee, by the data protection officer at the University of Oslo, and it is all linked and delivered to us in an anonymous way and stored at TSD, as you heard about previously. So this is really what we will be working for and with. And of course, there is the data lag time because a pregnancy it lasts 9 months. So we are now currently in the process of the data being pulled out, and we will be working with this spring. And this is just some of the data from Norway up until July 2022, just to point out that we cannot do this in one country alone. So up until July 2020, there were 13 pregnant women in Norway that had delivered, that tested positive for SARS-CoV-2. There were no intrauterine or perinatal deaths. All of these kids were normal, had normal birth weight. There was no need to transfer them to intensive care units. All were breastfed. But of course, we cannot base recommendations and advice on so few numbers. So this is just a map showing all the European data sources that are being linked, and we're actually capitalizing on previous investments and projects like the ongoing Conception project, which is a project under the Innovative Medicines Initiative, and where we have been developing the platforms, the tools, the common data models, because you can imagine how heterogeneous data is across all these different countries. So if we are to work together, well, we need to know how to standardize and analyze these jointly. Okay, so just going back to the question that I started with, and of course, so this nurse, she lived in America, and currently the American College of Obstetricians and Gynecologists, they have the following statement. Oh, so before, maybe I can just stop there and ask Angelique, so what was the audience's preference?
There were 8 answers for C, which was a yes, and none for A and B.
Really? So that's really interesting, because that's so the audience said, well, you should actually take the second dose, which is in accordance to what the American College of Obstetricians and Gynecologists say. You should take the second dose if you've taken the first. It's actually, when I did this with my students, they said, the large majority said option A, which is not to take the second dose, but actually, so the audience is very knowledgeable and in line. Actually, Folkehälsa, they're a little bit vaguer, so I apologize for those of you not able to read Norwegian. This is what currently, and please bear in mind that this is being updated as new evidence comes along, but so this is updated a couple of days ago. And the current recommendations are that vaccination of pregnant women, it should be evaluated on an individual base level. It's not contraindicated. And if a woman becomes pregnant during the vaccination period, well, here they say that the second dose should be postponed. However, they also open for that the second dose can be given in pregnancy after thorough consideration. And of course, one of the things that we're also monitoring is adverse reaction. This is just a list of potential adverse events of special interest related to the COVID-19 vaccines. And there has been several interesting signals already discussed. And of course, this is also something that we do in collaboration with the rest of Europe. This is just a picture of my dear colleague at the University of Oslo. And we are, our research group are members of the VAC4EU consortium, and where this is one of the missions of this consortium. And I think that in the sake of time, I think I will stop there. And if you're interested in more information about our research, please go onto our website. We published the first COVID-19 study on pregnancy just last week, and you can read more about it there. So thank you.
Thank you very much, Hedvig. This is, of course, very important. Pregnancy is a difficult case to study because for obvious reasons, and it's great that you have been able to establish this platform with the collaboration with all these different countries. So maybe Angelique, we have time for one question from the audience, if there is one.
No, there aren't any questions from the audience so far.
Okay, I thank you, Hedvig, because we have sort of limited time. So thank you very much, and you asked a question that you got an answer to. Okay, and then we go on to